Hi, everybody. It's been a little while, so welcome back to the Choroideremia Research Foundation webinar series. We're going to be having some additional webinars coming up soon. Our executive director, Kathy Wagner, has found some great contacts when she and several others went to ARVO this last year, so keep your eyes on your email. We'll be having some more. But our first is Katy Linkens. She's a new face to us, although she works with a face and a name that's very familiar to our organization, Maria Musagi in England. So just a quick couple things. Again, welcome everyone for coming this afternoon. We want to thank our sponsor, 4DMT, for being a part of this process, part of our webinars, and we greatly appreciate their support. We are recording this, so as always, I will be monitoring the questions and answers. Katy said she's okay with asking the questions at the time, so I will keep an eye on that, and I'll gracefully interrupt you, Katy, as you're going through your slides, and we'll also have questions at the end too. So Katy and I talked yesterday, kind of nailing some of these details out. And so I asked her for a description a little bit about herself. So I'm going to actually read it. I don't have it memorized. I apologize. But as I said, Katy is working you're close to the end, your final year of your PhD in working on stem cells with Dr. Maria Musagi. Her project is focusing on choroideremia, using stem cell models to understand the disease mechanisms and advance a non-viral gene therapy strategy. And I always like to ask, we always like to know the skinny on our researchers and people. We always like to know what's something for fun that you do. And so outside the lab, she enjoys hiking in the British countryside. I would love that. Including in the Peak District and Surrey Hills. That sounds enchanting. And attending musical theater shows in London. That's awesome. Most recently, she enjoyed a theater adaptation of the studio film My Neighbor Totoro.
Totoro.
Say that again.
It's Totoro.
Oh,
It's a
Totoro.
Japanese movie, I think.
Wow. That's perfect. Well, we are super duper excited that you're here with us.
Thank you. I'm really happy to be here as well. Thank you for asking me.
Absolutely. Do you know what I'm going to do? I'm going to go ahead and stop my, shut down my video. I'll pull things up here and you go ahead and you can begin. Welcome. Thank you again.
No, thank you for having me. Yeah, I'm really happy to be here. So thank you so much. I will just share my screen. There you go. Hopefully you can see that now.
We sure can.
Perfect, thank you. Yeah, so I'm Katy Linkens, as I've just been introduced, and I'm doing a PhD on choroideremia. My project kind of has two halves. So one half is kind of understanding the disease mechanism and what exactly is causing the disease, and the other half is advancing a non-viral gene therapy with Maria. But today I'm just going to talk about the first half. So it's like understanding the disease mechanism. And in particular, I've been looking at patterns of gene expression in choroideremia cells that I'll explain as we go on. And I'm sure I don't need to explain this on this call, but just to refresh my own memory, a little bit of background about choroideremia. It's an X-linked retinal degenerative condition. On the right, I've got some of the scans that Maria has taken in her clinics of choroideremia patients at the back of the eye. It's a rare condition, so around 1 in 50,000 to 100,000 people have this condition, and it shows a progressive degeneration of several tissue layers in the retina, including the RPE, photoreceptors, and choroid, and it's caused by mutations in the CHM gene. Sometimes I refer to this as the CHUM gene, just for shorthand. So as I said, I'm really lab-based. So what really interests me is all the different mechanisms within the cell. So on this slide, it's just a summary of the actual mechanism as far as we understand it. On the right is just a cartoon kind of depicting the function of the CHM gene. So CHM encodes a protein that's called RAB escort protein 1, REP1. And what REP1 does is it binds to a set of proteins which are called RABs. And it binds to them as they're being synthesized. And then it attaches a lipid tag. So it's like a little fatty group called a prenull group. So it attaches a prenull group to the RABs. And then it stays bound to the RABs until it has trafficked them, transported them to their target membrane. And then the lipid group that it's attached really helps it to embed into the membrane. So REP1 is really indirectly responsible for regulating trafficking throughout the cell, or like the transport of different RAB proteins. And RAB proteins themselves carry different types of cargo around the cell. So it's a very important protein, REP1. It's expressed in every cell in our bodies. But the particular cell type that I'm interested in is called the RPE cell. So on my next slide, I have, again, a cartoon diagram kind of explaining the RPE cells. And RPE stands for retinal pigment epithelium. This is a tissue layer that sits in the retina at the back of the eye. It sits between the choroid blood supply and the photoreceptors, which are responsible for vision. And it's a very, very, very specialized tissue. The RPE, it consists of a very tightly packed monolayer of cells, and it has a lot of specific functions. Some of these include maintenance of the photoreceptors, so it keeps them very healthy. It expresses and produces melanin, which is a pigment responsible for absorbing light in the eye. So that's a very important function for protecting the retina, and also absorbing light scatter. So it helps us keep really fine detail in our sight. The RPE is very tightly packed, so the cells are very close together, and that helps to maintain a barrier between the blood supply and the retina and photoreceptors. And this is very important because it helps to regulate the transport of different substances in and out of the retina. These substances include water, ions, nutrients, and it's also responsible for the removal of waste from the retina into the blood supply. And another important function of the RPE is that it maintains what we call an immunosuppressive environment. So essentially what that means is it reduces the extent of inflammation and immune response in the retina. Because if this process is not tightly regulated, it can be quite destructive to the retina. And as the structure of the retina is destroyed, of course, our sight is lost. So these are some of the very important functions of the RPE tissue. And to carry out these very specialized functions, the RPE requires a very specialized shape, morphology, protein expression levels. And of course, to achieve all of this, it's very important that it expresses all the right genes at all the right times and all the right amounts. So essentially, for this project, I'm looking at gene expressions of the RPE, and if they're disrupted, how does that affect the function, and how can a disrupted function of the RPE then cause a disease phenotype? So yes, RPE are my cells of interest for this project. But on my next slide, I just have a diagram explaining how we get these cells in the lab, how we generate them. So on the top left of this slide, there's a cartoon of an arm, and that's to depict the biopsies that we take from choroideremia patients who are willing to be involved in the research that we do. So we get them into clinic, and Maria will take a skin biopsy, and these will convert into skin cells, which are also known as fibroblasts. And from these fibroblasts, fibroblasts in themselves, they can be used as a very helpful model in the lab. But what we can also do is turn them into stem cells or induced pluripotent stem cells. And these are even more helpful to us because these stem cells can be turned into any type of body cell. And so for our purposes, of course, we turn them into retinal cell types, specifically RPE. So I've got some pictures here. It's a bit small, but in the bottom middle section, that's a picture down the microscope of our iPS cells. So they grow in like this circular formation, but this is actually lots and lots of cells all tightly growing together, and they form a colony. And they typically grow in this very tightly packed with dense nuclei and a circular edge, essentially. So that's our stem cells. And then after we've converted them into the RPE retinal cells, they look like this. So this is the cells in a tube. We collect them and we spin them in a centrifuge to gather them all at the bottom in a pellet. And you can see they're very dark. And this is due to the melanin production. And this final image, that's again an image taken down the microscope. So you can see the cells are very tightly packed. They show this kind of hexagonal morphology. We call it cobblestone when they're all so tightly packed together in that kind of hexagonal shape. And again, they're very dark due to melanin production. So yeah, once we've generated these cells in our lab, we generate them both from choroideremia patients and from healthy individuals that have no retinal phenotype at all. So then we go a bit further and we start to characterize this RPE. And one of the first things that we do is we stain for different protein markers. So in this panel, you can see, again, images down the microscope after labeling for specific proteins. So in the top panel, this is a healthy individual, and we've stained for markers that we call ZO1 and MITF. So these are essential for RPE, healthy RPE function and generation. And then on the bottom, we have cells that we've generated from our choroideremia lines, and they also express ZO1 and MITF markers. And then in the blue, we have a DAPI stain, which is just a generic nuclei stain we do for all cells. So after we've done this analysis, we can see that we're producing quite a high quality RPE in the lab. It looks how we expect it to look, and it seems to be a good model. So after we've done this quality check, if you like, we take the cells further and we begin to study them even more in depth. And for this experiment, what I did is I grew the cells in the lab, I harvested the cells, and I extracted RNA. I sent that RNA for sequencing, and then we put it through some pipelines, so some computational analysis. And essentially, the data set we got from that allowed us to see exactly which genes these cells were expressing and to what level. And so we could begin to compare between the patient lines and the healthy lines, and we could see which genes are overexpressed and which genes are underexpressed, and which pathways are they involved in, to try and get an idea of which pathways are disrupted in the choroideremia cells we have in the lab. So on this next slide, this is what we call a heat map. So it's kind of a summary of the data set that we had. On the left, this refers to our choroideremia cells, and on the right, it refers to our healthy cells. And just to summarize what this means, all of these little lines in red represent a gene that is overexpressed in choroideremia cells, and all of the blue lines below represent a gene that is underexpressed in the choroideremia cells. So from this figure, we can tell that quite a lot of genes are dysregulated in choroideremia cells compared to our healthy controls. So there is a clear phenotype that our cells have. There are some clear differences, but what exactly are they? We need to go deeper in to understand precisely which genes are dysregulated, and again, which pathways are they involved in. So on my next slide, one of the first things that I could see from this data set was that markers of healthy RPE were dysregulated in the choroideremia model, the choroideremia cells. So you can see these blue boxes on the left depict that. So they show all of the different genes that are downregulated, underexpressed in choroideremia cells. And a lot of these are what we would normally consider RPE markers. So we could see from our previous analysis, because the shape and the color and everything was looking okay, was looking good for ourselves. So we knew that it was a good quality RPE model that we had. But once we go into actually quantifying the gene expression, we can see that some of the markers needed, some of the genes needed for a healthy RPE are underexpressed. So it begins to suggest to us that the RPE in choroideremia is functioning kind of suboptimally. And on the right of this figure, this is a list of all the genes that are affected specifically. And this red box that I have has highlighted a gene that was very interesting to me. That gene is called Best1. And it appears from this data set that Best1 is severely downregulated in choroideremia cells. This was really interesting to me, as I'll show on my next slide. because Best1 is a chloride ion channel, and it's found on the basal bottom side of the RPE. But it's very interesting because mutations in this gene actually cause another retinal degenerative condition or set of conditions called bestrophinopathies. So these are retinal degeneration, which the clinical phenotype does show some overlap with choroideremia, I believe. So this is something that I'm looking into further to really understand what might be the similarities between these two conditions, if anything, and to try and understand if there are some shared pathways that are disrupted here. So this is something that's ongoing at the moment, and I'm looking into further. Another thing that I saw in this data set is that the RAB genes were dysregulated. So some of these RAB genes are upregulated in choroideremia cells and some are downregulated. And yes, just referring back to the earlier slide, RABs are very important for transporting different types of cargo around the cell. And REP1 itself is important in guiding this transport system. So previously from the literature, we did know that REP1 altered or it had a different rate of prenylation of the RAB genes at a protein level. But what we didn't know is that in choroideremia cells, we could see that the genes themselves were dysregulated as well. And when I began to look into the function of these RAB genes, I could see that a lot of them were involved in melanogenesis. And just moving to the next slide, this begins to suggest that the melanogenesis pathway is disrupted in choroideremia cells. This is something that we've seen in other models. So this panel on the right-hand side is a published data set from the zebrafish choroideremia model. And you can see that compared to the wild-type healthy zebrafish, they have quite a high amount of melanosomes, of melanin vesicles, and they're very structured. It's very orderly in retinal layers, as we would expect. But on the right, the choroideremia model shows a loss of melanin and a disruption. So the melanin vesicles, which we call melanosomes, are a lot smaller. They're a lot more dispersed throughout the layers. And just in general, there's a lot less melanin. So it's very interesting that we can see this reflected in our cell model as well. This image on the left, again, this is showing RPE cells that we have grown in the lab and harvested into tubes. And in the left tube, you can see a wild-type healthy cell pellet, and it's a very dark color. On the right-hand side is the choroideremia cell pellet, and there is still pigment, which is really good, but it is lighter, so it suggests that there's less pigment. It shows more of a gray color than a darker color. So yes, again, this was something that I thought was very interesting and something that warrants kind of further investigation, is the disruption of the melanogenesis pathway appearing in our iPSC models. Another phenotype that I could see was a disruption of inflammatory pathways. So earlier we were discussing how important it is that the retina really controls any inflammation that might happen or any immune response, because immune responses are important to keep us healthy. But if an immune response is uncontrolled, it can become quite destructive. And the eye is a very sensitive and specialized tissue, of course. So any kind of uncontrolled inflammation is going to cause disruption to the cell layers in the retina, which are essential for sight. So I could see kind of an upregulation of a few different inflammatory pathways, including inflammasome upregulation. That's very interesting because in AMD, which is age-related macular degeneration, and that's the most common cause of the loss of sight in adults. Inflammasomes are also dysregulated, so we can see some overlap between choroideremia and AMD in that respect. And we also went on to do some pathway analysis. So we were not just analyzing which genes are disrupted, we were really looking at which pathways are these genes involved in. And I could see that there's an upregulation and increase of diseases relating to cell death. There's an upregulation in chemokine signaling, which are like immune response molecules. And there's also a disruption to cell adhesion. So that's suggesting that the barriers or the contact points that really hold the RPE cells very tightly together to maintain the barrier, those seem to be breaking down as well. So that was a lot of information, but just a summary figure of the inflammation pathways that I've just spoken about. In this figure, you can see on the left a healthy retina, which has like intact barriers, and any immune responses are very tightly controlled. On the right shows a retina where the barriers between the RPE are breaking down. There's an upregulation of different immune molecules, such as cytokines and chemokines. And there's also a migration of immune cells from the bloodstream into the retina, which is generally considered quite a detrimental thing to the retina. So these are all results that we're seeing from our analysis of the gene expression, but it's also matching up to what we've seen in the literature, because from different autopsy histology analysis from patient eyes has shown that different immune cells do migrate into the retina in choroideremia, in some choroideremia patients. And it appears that that's matching up with what we're seeing in this gene expression analysis. And the final phenotype that I would like to show you from this data set is called a senescent phenotype. And senescence is essentially, it's a cell that is aging prematurely. And essentially, it's often due to like DNA damage in the cell. So because there is some kind of DNA damage in the cell, it essentially stops itself from growing, it stops itself from replicating to prevent passing on this DNA damage to future cells. So senescence is when a cell stops growing, it's aging earlier than it should do, and it also starts producing signals to other cells around that they should start doing the same thing. So it just promotes an environment where cells stop growing and stop dividing, and it also promotes an environment that again is inflammatory. So a senescent cell will secrete, it will release a lot of molecules that are inflammatory. So from these two heat maps, again, you can see the red panels indicate an upregulation, increase in genes that are associated to this senescent or aging phenotype. And on the right, this heat map refers to the secretory phenotype, so the release of molecules that are inflammatory. And again, when we went further into analyzing which pathways could be implicated in this, we saw there was an upregulation in pathways that are associated to genomic stress and DNA damage. And also oxidative stress was coming up as well, which is something, again, that we know is a phenotype of choroideremia cells. So this is just this slide is just a cartoon representation of the senescent phenotype and why it's so important. So as I was trying to explain, the cell releases molecules that are pro-inflammatory. So it starts to call immune cells towards itself, and it starts to promote other cells to also enter into this this arrested state, this state where they stop growing and they are calling inflammatory cells. And again, this phenotype is one of premature aging, which is quite interesting because choroideremia retinas in general, often the disease phenotype comes from signs of premature aging. So again, it's quite interesting that what we are seeing in the cells is actually matching up to what we are seeing in patients in the clinic. And again, senescence itself has been shown recently to be implicated in AMD, which is really because AMD is so common in people. There's a lot of research and a lot of time, a lot of money going into diseases such as AMD. So it's actually very interesting when we can find underlying mechanisms that show a similarity between rarer disease such as choroideremia and more common diseases such as AMD. Because then we can really use all the research that's been doing in AMD to give us further insight into choroideremia. And also, if people start to explore new treatment strategies for AMD, there's potential for these to be applied to rare diseases such as choroideremia, if there is a commonality indeed in the mechanism. So that's the end of the talk, but just some take-home messages and summary. The iPSC-RPE that I've grown from choroideremia shows a disruption in melanogenesis, so pigment production. It shows a pro-inflammatory microenvironment, which is not normally seen in the retina. And it also shows a cellular stress-induced state of senescence or premature aging for the cells. So these are the key pathways that I'm seeing in this data set for the choroideremia cells. And this is all really interesting because some of these pathways are quite novel, novelly linked to choroideremia, that is. So it kind of paves the way for new therapeutic strategies, possibly. For example, the senescent state that I was just mentioning. In AMD, there is some discussion and some very early research where people are trying to apply drugs targeting the senescent pathway. So they call them xenomorphic drugs, and then it stops or prevents from spreading the senescent phenotype. So potentially these are approaches that could be considered in the future for choroideremia. And the main next step for this part of my project is really functional validation of this data set. So we've been studying these cells on a DNA level. The next step is really to see how does this actually affect protein expression, and further from that, how does this actually affect the function of our cells? We can see visually some functional validation, such as the loss of pigment, but there's further studies to do just to confirm all of the pathways I've just mentioned are actually disrupted at a functional level. And that's all, but I would like to say a big thank you to my lab and to Maria, of course, and to all the patients who make our work possible. And thank you for listening as well. Happy to take any questions if there are any.
That's wonderful. Thank you so much. I'm going to keep my video off. That was wonderful. You explained very well, a very complicated process. I think you did that very well. Well done. We did have somebody ask the following question. Thanks for the great talk. Does the aging phenotype can be seen in RPE cells affected with CHM in vivo?
Oh, that's a really good question. From what I know, I don't work with in vivo models, but from what I know, the premature aging in general is seen in the in vivo retina, but specifically the senescent phenotype, I don't think anyone has checked yet. This is something quite new that's coming up, I think. I haven't actually seen it published anywhere, although others have discussed it with me face to face. But I think it would be really interesting to check that. Yeah, it's one of my next steps to really confirm it in the cells. So I think it would be even better to confirm it in the zebrafish and the mouse models as well.
Another question. Did I see on the slide that there is an increase in VEGF and CHM? Would CHM patients benefit from VEGF inhibitor therapeutics in development for AMD?
Yes, you did see that. That is something that I'm trying to understand further because there's different types of VEGF. So I think it's VEGF A, B, and C, and not all of them are increased in this data set. So I did look into that as well, and I had the same thought as you, but I think it's unclear at the moment. I think it's too early to say. And I also know that in published papers, I think people have checked the VEGF levels and they seemed fairly normal. So I'm not sure that it would be the best route to follow on the data that we have so far, but definitely worth investigating more.
Similarly, was there evidence of complement cascade in CHM? If so, could CHM patients benefit from C3 and C5 inhibitors that are approved for AMD? So before you answer that, what does the question mean exactly? If you could help us understand the question too.
Yes, yes. So the complement cascade, that's a very important inflammatory pathway. And I think what this person is referring to is this is being targeted for other conditions. So I think there are gene therapies in the pipeline that are trying to target this inflammatory pathway. And I think, if I'm correct, it's to kind of dampen the immune response. So to kind of dampen the inflammation in the retina and preserve as much of the structure as possible. And it's a really good question. I did look into the complement pathway, this specific inflammation pathway, to see if it was disrupted in this data set. And again, I couldn't see clearly that it was. Some elements were upregulated, a few were downregulated, but there wasn't a clear, absolute disruption to the pathway that I could see. And again, I think at a protein level, people have checked in various cell models the complement pathway, and there's nothing conclusive. Again, it's definitely something to keep an eye out for.
The cells that you used, Katy, were those in carrier women or were they affected males?
So it's all affected males. All of our choroideremia lines are affected males. But yeah, it's a really good point. It's very important to check the female carrier lines as well. This is an issue in science research that I hadn't come across so much until recently, and it's the fact that so many of our lines in the lab are male, not just for choroideremia, just in general. So we're starting to really gear towards pushing for more female lines as much as possible. So that's a really good question. Thank you.
We do have quite a few women that I'm presuming are carriers, such as myself. Do you have any other thoughts on, or will you be using some of this work with carriers? Is Maria or others that you're working with kind of branching out into the carrier studies?
As far as I know, we're not branching there at the moment. I would say, yeah, but it is very important. So thank you for highlighting it. And I think I need to go back and consider this data set in that context. So thank you for highlighting that.
Sure. Do you know what? That's all the questions that we have. You were very thorough.
Thank you.
Let's just give
They're
it one
very
more
good
minute.
questions, though, maybe.
If anyone has any other questions, go ahead and feel free to type them in. Oop, here comes one. Oh, it's just thank you. Kathy says thank you.
Oh, thank you. No, thank you for having me. I'm happy to be here. So.
You know, it's really encouraging for a person such as myself who's been in the organization since 2006. It's really wonderful to have new, young, vibrant researchers coming on board and specifically working on choroideremia. So thank you. Thank you to you and a lot of your colleagues for working on this. And here's a couple more comments. Let me just get it. Thank you for your dedication, Katy. Is this approach a potential for restorative or stopping further degeneration?
So I think it would be the latter, so stopping further degeneration. I think the restorative, as far as I'm aware, the best options we have are like the cell transplants. But all of these disrupted pathways, I think it would be preventative measures, really.
Which is still a winner for us. And we have another person saying, thank you very much for your presentation. Wonderful. Here comes something else. Thank you guys for your patience. Oh, again, thank you. Another thank you. Perfect. Well, Katy, thank you. Thanks to everybody for, oh, and you know what? I found the answer, Katy. It's October 12th. We are going to be in London. I asked Corey to verify and he said October 12th.
Perfect. I'll write that down.
Good.
Nice.
Okay, here comes, we have another question. Just the question is, why no viral vector?
So that's just our approach. Our approach is we're really focusing on non-viral vectors. There are labs that are still going for the viral approach. The reasoning behind our approach is that from other approved viral therapies, so for instance, Luxturna, it's coming apparent that some patients are showing inflammatory responses to the therapy. And as far as I'm aware, these responses haven't yet caused sight loss, but we're not sure what will happen in the future because it's very early days. So the idea with the non-viral is that because it has no viral components at all, it will not induce inflammation in the eye. And then we will not see these damaging inflammatory responses, which is especially important for conditions such as choroideremia. If we know the retina is already in a pro-inflammatory state, we don't want to exacerbate that.
Someone asked if that would be Dr. Harbottle.
Doing the non-viral.
Mm-hmm.
Yes, so we have collaborations with Richard, yeah.
Mm-hmm.
And others as well, but yeah, he's a main player in that.
There's little snippets coming in on this thread, Katy. So how will you transport following up after the no viral vector?
Sorry, could you repeat that?
Oh, how will you transport is the question. It was a follow-up question to the same person is asking why no viral vector, which you explained, and then how will you transport?
I see now I understand. So there's different vectors you can try. There's different types of nanoparticles. So at the moment we don't have a clear option. We're investigating a lot of different options at once. But there are different nanoparticles around that lots of people are trying. So we have a few collaborations ongoing, but we're not quite ready to present that just yet, I'm afraid. But it is ongoing for sure. And yeah, when I publish my thesis, you'll have all the details.
And when will that be?
So I finish April next year.
Oof, are you counting down the days?
No, I wish I had more time to get everything done. I feel this project it's so exciting. There's so much to do. Just hope I can do it justice. But it's very interesting to work on, so I enjoy it.
Wonderful. Well, we thank you. Thank you for your time, and your energy and your compassion and your commitment. Thank you.
No, thank you for having me. I'm happy again to be here.
Good. Enjoy the rest of your summer. And to everyone else, join us next week. We are going to be having another webinar. So please come, and we greatly look forward to seeing you next time. Again, thanks to 4DMT and to Katy. We'll see you again soon, everybody. Have a great weekend. Bye-bye.